Well, thank you very much, Lucia. And thank you also for inviting me and giving me the opportunity to talk a little bit about the research we do. As mentioned, the title of my talk is Plasmonic Biosensors for Biomedical Applications. I think I have to warn you that it's going to be much less plasmonic than the majority of the talks you have heard so far. Instead, I will try to take you on a journey to see what all different kind of challenges you may encounter when you try to bring your plasmonic structures or nanostructures into sensing or biosensing applications. But before I do that, let me start with a little introduction and wrong direction. Okay, maybe starting with the brief definition of what is a biosensor. According to International Society of Analytical Chemists, a biosensor is an integrated device which consists of a receptor and transducer that is able to provide selective and quantitative analytical information using a biological recognition element. Of course, this might sound not telling much, but if we use an example, handheld glucose monitoring device, I think it will be more than clear of what we are talking about. These glucose monitoring devices have been around for several decades, but it might be nice to remind ourselves that it all started in 50s and 60s, which also illustrates how long it takes to bring technology from the fundamental concept to useful medical device. Here are just on this slide, there are just few examples of, okay, wrong direction again. Devices, this is the first commercial device. And I'm sure you have seen devices like this before. You basically prick your finger, take a little blood droplet and put it on a surface of this little strip where it leads it to the device where there is electrochemical sensing device and provides immediate readout. Of course, over years, the technology has advanced tremendously, and today we can also, or people with diabetes can also use devices which allow for continuous monitoring, not from blood, but from interstitial liquids, which of course is a lot more convenient and nearly painless. So another thing that I would like to highlight here is the level of analytes these glucose monitoring devices are typically measuring. As you see here, we are talking millimolar or milligram per mil, if you prefer these units. And I would like that you keep this in mind when we talk about some of the applications later on. Of course, these are not only analytes like glucose to be detected. In fact, there is a whole universe of other things which biosensors can potentially target, whether these are biomarkers of diseases or health, whether these are contaminants and pollutants for our protection of environment. Also, food safety is another important application where detection of foodborne pathogens and toxins becomes a very important issue. And of course, biological and chemical warfare agents are also targeted by biosensor technology. It's not only though about detecting the biomolecules, also there is a lot of interest in understanding biomolecules and biomolecular interactions and relate these molecules and processes or interactions among them with the diseases, their onset or development. And here biosensors can provide important questions on what are the molecular mechanisms behind diseases, or whether these pathways or interactions can be intercepted, leading to new diagnostic or treatment modalities. What are the main challenges for development of biosensors for medicine? We were talking about glucose levels, but if you try to look at some of the markers, especially related to interesting or severe diseases such as cancers or Alzheimer's and others, you will find that the analytes you want to detect are present at extremely low concentrations. If we used to say, and I think it's probability-wise about correct for glucose monitoring, that we were trying to find a needle in a haystack with femtogram or range of femtograms to nanograms. We are talking about 10 to the 6 to 10 to the 9th haystack. That's a lot of hay, as you can imagine, and requires that the sensor technology is not only very sensitive, but also very specific. So this is the first challenge. Clearly, another challenge is that biological media are very complex. While there are small concentrations of the target molecules we would like to detect, there are at the same time, there is basically abundance of the molecules which may interfere with the measurements. And this effect is often referred to as non-specific adsorption, where non-target molecules get attached to the surface, producing false positive sensor response. Another issue is the composition of samples. If we go and work with samples such as blood or cerebrospinal fluid, it is highly variable. It's not only variable from patient to patient, but it also varies during a day for the same patient. So it's very difficult to find a reference, and this variability also makes it quite challenging, and requires that the technology is really robust and stable to be able to deliver the performance you need for medical applications. So as was mentioned earlier, I'm going to talk about affinity biosensors based on surface plasmons, which are basically optical biosensors that employ optically excited surface plasmons to probe the molecular interactions between the receptor molecule and analyte typically present in a liquid sample. I think this picture, and I'm sure most of you have seen it many times, illustrates basically all the fundamentals related to plasmonic affinity biosensing. And from that, you can quickly deduce that it's direct, real-time, doesn't require any labels, in principle is quite sensitive. And also it's another important thing, it's non-invasive in terms of requirements of the molecules or interference with the molecules you try to work with, try to detect. But also from this picture, I think it becomes clear that you have to take care of a lot of things, and you have to bring together a lot of pieces of technology from different fields to make the biosensors functional. Of course, at this conference, there was a lot of discussion, and there's going to be a lot of discussions about plasmonic structures and nanostructures. But you also have to have optical platforms, which are basically allowing you to excite surface plasmons and read information from surface plasmons. Microfluidic systems or devices, which are defining the conditions for the molecular interactions and providing additional sample handling capabilities. Functional coatings, that's another important aspect. You have to be able to deliver the specificity to the sensor. You have to combine with appropriate receptors, which need to be attached to the surface in the form of functional coatings. That's only aspect I'm not going to talk about today for the time constraints, but we'll be happy to take any questions afterwards. And of course, you have to have appropriate assay or detection formats, which are appropriate for given application and provide the performance you want to have for that application. So, few words about plasmonic nanostructures, and already there has been so much said about plasmonic nanostructures. I'm not going to go into details. I will just mention that in the beginning, there were basically two main types of plasmonic structures and surface plasmons, which were being used for sensing, so-called propagating surface plasmons, propagating basically the interface between a metal and a dielectric, and localized surface plasmons, which were unlike the propagating ones, not propagating, but localized to various nanoscopic particles or patterns of particles. Important thing for sensing here to mention is a typical field extent, while the propagating surface plasmons have quite a large field extent into the medium which is being probed. This is not true, or it's quite different for localized surface plasmons, which typically provide the penetration depth about the order of magnitude less. And that needs to be taken into account when you are designing the sensor for a specific application. Right, of course, over the last 2 decades, there have been a lot of development in terms of nanofabrication and also computational capabilities, allowing to design and develop more and more sophisticated structures. And here are just a few examples, and over the conference time, we have seen many more. Of course, these structures are wonderful in terms of delivering the electromagnetic field enhancement to the specific areas where you want to have it. Also allows to help with field distribution and associated with analyte transport. On the other hand, they are still challenged by, I would say, lack of cost-effective chip manufacturing. Their benefits are often related to your ability to functionalize them locally, which is another thing you would like to have, and it's quite difficult. And of course, to make a biosensor, you have to make them robust enough and integrate them into optofluidic structures and chips. In the sensing community, there have been a lot of discussion about how to tell which structure, which nanostructure is better for sensing. And I'm sure most of you have encountered those discussions or participated in them, especially for people who are doing theoretical simulation. Often the simple answer is to use the figure of merit, which is basically a sensitivity to bulk refractive index change over a width of a plasmonic feature. And although I think we all kind of understand that this is probably not the whole truth, it has been used for many years like this. Therefore, after a certain time being not happy with that, we started a project in which we decided to basically analyze the relationship between the features of plasmonic structures and performance of a biosensor which uses such structures. And I'm not going to go into details. You can find it in the paper we published last year, but we showed that there can be expressed analytical solution for refractive index resolution, which is well corresponding to the limit of detection, for example, in sensing world, which is related to the capabilities of the optoelectronic part of the system and the part which is related to the spectral features of the nanostructure. Here you clearly see that the figure of merit is in this old form is present and it is relevant, but though it is not the whole truth, as you see, there are also other parameters which need to be taken into account. This is why we decided we are going to propose a new figure of merit, and also in this paper, which is stated right there, we showed based on silico experiments that the model correctly describes the behavior of biosensors based on plasmonic nanostructures and standard optoelectronic components. Right, so this was a little bit about nanostructures, and I think the main message was maybe not look at figure of merit as traditional literature says, but try to look at how the nanostructures work with your specific optoelectronic system, and then there are models which help you predict what performance you can achieve. Another important component of your biosensor is an optical system, and I'm going to show you just a couple of examples of optical platforms we built in our lab over years. This is a system for spectroscopy of surface plasmons. As you see here, it's basically based on sub-ops on a correction geometry of attenuated total reflection and wavelength spectroscopy. So nothing major. We just put a lot of effort into optical design and making sure that it has temperature stabilization, right software, etc., and provides resolution near to 10 to the minus 8 refractive index units. This platform not only allows us to work with chips with continuous films and propagating surface plasmons, but also with other structures, which is something, and this is just one example of structures we have done before, array of gold nanostripes. And the reason why we also like to have one optical system to be able to address different structures is for comparison. And I'm sure you have seen it as well, that there are a lot of papers in literature where the results are compared to standards which are not correctly specified or are, let's say, iffy. And therefore, what we really like to do is to perform all the side-to-side comparison, but we do something on other structures to also do the same thing on continuous films to have a strong reference. Another type of structure which allows basically the same thing, having continuous surfaces as a reference, is the imaging system. This is the imaging system with normal incidence and collinear beam, and it's compatible with diffraction rating as well as arrays of plasmonic nano-objects. Its performance is slightly worse, but at the same time, it offers a lot more information because of spatial readout. So definitely useful for measuring arrays or detecting large number of interactions at the same time. We also have developed a variety of compact field devices in the effort to bring the plasmonic technology to analytical applications in the field, but for time reasons, I'm not going to cover that, just want to mention it. Another important aspect is how to handle the fluid which contains the analyte molecules which are supposed to bind to receptors on the surface. Here, if I had time, I would be happy to talk about mixing structures and how to best bring the analyte molecules to the sending surface. But because this is a conference about nano optics and nanoplasmonics, I'm going to select one topic which is somewhat different, and it's about comparison on how different densities of plasmonic structures or nanostructures are affecting the performance of the sensor or biosensor based on this. I think I do not have to explain that to have a biosensor, you typically try to interface your nanostructure with the liquid sample contained in some flow-through flow cell, which is illustrated right here. And this is what happens at the sending surface. These ion nanoparticles with receptors, they capture the analyte molecules, and this is the schematic of the flow as it flows, as the sample flows through the flow cell. As we probably all know, nanostructures with low fill fraction, meaning low density of nanoparticles, exhibit better analyte transport, but at the same time exhibit worse optical performance. So clearly there is going to be some trade-off which is going to provide best performance, or this is what we thought when we embarked on the study, when we tried to combine the optical performance as well as mass transport performance to come up with a question, with the answer for what nanostructures work best in terms of mass transport as well as optical performance. And we again developed an analytical model which allows to correlate minimum detectable concentration with elements or factors of parameters of the device. And again, I'm not going to go much into detail because it's all described in the papers we published, but it incorporates both the optical performance expressed in terms of sensitivity as standard deviation of sensor output, as well as mass transport efficiency, which is in part related to diffusion limited mass transport, as well as binding characteristics of the two molecules, the analyte and receptor on the surface. To experimentally demonstrate that there indeed is a benefit of smartly playing with mass transport and optical performance, we carried out an experiment in which we developed a biosensor based on an array of gold nanorods. As you see here, we prepared sensors based on different field fractions. From very low density to quite high. And of course, we also had as a reference standard SPR sensor that's continuous surface. So what you should notice here is that when you have relatively high concentration of analyte molecules binding to the different substrate, there is not really much happening. But when you go to small concentration, you start seeing the difference, and you see the structure with lowest fill fraction performing or responding fastest and providing largest response because of the mass transport improvement because of the low density. We did the detection experiments and measured the concentrations or determined the calibration curve and determined the lowest detectable concentration by experiment and compared it with the theory. And you see here that the experiment and theory both very similar in terms of there indeed is an optimum fill fraction at which the lowest concentration can be detected. And while there was a quite a good agreement, despite that there was the theoretical and experimental values were a little bit offset. And the total improvement was about order of magnitude, and I think depends on structure, the receptors, and so on. But it shows that indeed, plasmonic nanostructures can provide, if you design them smartly, having in mind both optical as well as microfluidic properties or mass transport properties, can provide considerable improvement over SPR centers. Now I'm going to talk about two applications. I selected applications which are, in my opinion, fun because we did them in both in kind of contra-intuitive way, almost in opposite way as most people do them, and they both led to interesting results. They are both related to myelodysplastic syndromes, which is a group of oncohematological malignancies. It's quite complex disease, and the final stage of the disease to which the disease may progress is called acute myeloid leukemia, which is a severe health condition, and it requires extensive treatment. So we will start with a first example, and this is a detection of protein interactions related to MDS. This is something what we have been thinking about for quite some time, whether if we do things differently than most people do, meaning if instead of putting receptors on a surface and detecting the target molecules, if and what would we see if we instead just looked at how the blood plasma from different patients interacts with the surface of the sensor covered with variety of different molecules, variety of different proteins. And for a long time, we thought that this was not going to be telling us much, but eventually we decided to try it, and we used SPI imaging sensor to do that, and I'm not going to go into details of the experiment. But what would like to quickly go to the data which showed result of small clinical study where we had about 80 blood plasma samples from different MDS subgroups, including also the patients with acute myeloid leukemia and healthy controls. And you see that there were 6 different proteins, and that some of them were consistently responding differently to different subgroups, highlighting that indeed some of them might be an interesting way to distinguish or discriminate the stages of the disease in future. So in this case, I would like to say that we don't know what specifically we are detecting. We are detecting something from blood plasma. Of course, we are curious people. So once we performed this, we did want to learn what was on a chip. So we took the chip and eluted the proteins from the chip and took them from mass spec analysis. So we learned. Also, there is interesting aspect which how we actually want to and continue to improve this idea that we take proteins which were identified through mass spec and putting them back on a chip, and in this case extend or improve the capability of this interactinic approach. So this is indeed not detection of biomarkers, but rather broadband interaction analysis of proteins with the samples of blood plasma patients. Second application is related to the detection of microRNA. As you know, microRNA has emerged as a very important medical diagnostic paradigm and has been linked to a variety of diseases, including MDS. For MDS, there have been quite a few, and we especially work on the microRNA 16 and the 125. And if you look at the left side of this picture, this would remind you the standard sandwich assay most people do very often. You basically have a surface with your receptors, you capture the target molecule and then incubate the surface with the nanoparticle linked with the complementary sequence so that it binds to the part of the receptor, of the analyte molecule or DNA or microRNA sticking out and basically enhances the sensor response. Well, as we have been doing this for quite some time, we learned that the performance of such a biosensor is basically, at the end of the day, limited by how variable is the background, which is basically a binding of functionalized nanoparticles to the reference surface. And as you see here, these are basically two responses from two different reference channels prepared on the same chip in identical fashion. So this is as close as we get normally. And you see that there has been quite a variability. And as this was limiting the performance, we figured out that maybe we would like to do differently. And instead of measuring the capture of the molecules or of nanoparticles, we said that we are going to be looking into release of nanoparticles instead. So we first capture them and then come up with a special DNA which knocks them up, right? So they get released, and you see here a response of such a sensor to surface, which was incubated with 150 femtomolar concentration of this microRNA, and you see nice release. And reference channel where you basically see nothing, and you also see basically no variability of the sensor response. This is about 2 orders of magnitude, which is exactly what you eventually get when you compare the performance of these two assays. This is just for comparison direct detection, which is not all that interesting at this point, as this is very difficult to do in complex samples such as blood plasma. And here you see sandwich assay. These are the green points, which goes down to about 28 femtomolar, which is not bad a limit of detection. But if you look at nanoparticle release assay, how far it can go basically below 1 femtomolar to about 250 attomolar. What is really nice is that this brings the performance of the biosensor to about an optical noise in the system again. So long time, we have a multidisciplinary group, and long time our physicists were saying to our chemists and biologists that the problem of performance is their problem because the optics works wonderful and chemistry is not. With this assay, we get again the ball a little bit more on the side of physicists, and our chemists come back and say, you have to improve your optics again. So, which I think they did. And it has not been published yet, but I think in the latest experiments, we were able to get about 10-fold improvement, getting us somewhere to 50 attomolar concentration. This is this number is in blood plasma, which is important, and it makes this approach comparable to the PCR, which is a gold standard for detection of microRNA. But what is really important here is that unlike in PCR, where you have the extraction, you have transcription, there are a lot of steps which require technology and people. Here, there is basically no sample prep. It is basically blood plasma diluted and analyzed by the sensor. And you still can get this level of performance. Well, I think with that, I would like to come to conclusions of my talk. I hope that I convinced you that there have been some interesting advances, not only in plasmonic nanostructures, but also in optics, microfluidics, and detection methodologies, and that they have expanded the performance and capabilities and potentially applications of plasmonic biosensors. It should be stressed that non-specific absorption remains a main challenge for deployment of plasmonic biosensors or label-free plasmonic biosensors for analysis of complex media. Though there have been advances on fronts of functional coatings as well, and if you look into literature, you see more and more papers, and a lot of them are quite satisfactory, where people are demonstrating applications of plasmonic biosensors in medical field. I hope I showed you two interesting ones, which, as I said, are somewhat different from the mainstream, but for the mainstream, you can read the reviews and get, I think, a lot more balanced picture. With that, I would like to thank my colleagues and students and collaborators, and of course, would like to thank you for your attention, and we'll be happy to take any questions you might have.